Welcome to our business tract of the Data Science Salon YouTube channel. This presentation will highlight various data science strategies applied to the management and analysis of large-scale cancer data sets with the goal of facilitating innovative data-driven research and developing novel efficacious therapies. Vasileios Stathias, lead data scientist at Sylvester Comprehensive Cancer Center, goes in depth with various strategies applied. Enjoy.
Hello everyone. First I would like to thank the organizers for giving me the opportunity to present our work here at the Data Science Salon. During my talk I will be sharing with you data science and artificial intelligence approaches that we are leveraging at the Sylvester Comprehensive Cancer Center and at the University of Miami to accelerate cancer research and identify new cancer targets and compounds. On the first part of my talk, I will go over our newly developed data management and analytics platform that we're implementing at Sylvester. And using this platform, researchers are going to be able to access, analyze, explore, and share their next generation sequencing and biomedical and multi-omics datasets. And this way, they can further advance the data-driven research. On the second part of my talk, I will be going over a few examples of how, once we have these well-curated and annotated datasets, we can utilize machine and deep learning algorithms to identify new compounds, targets, and therapies. As we know, cancer is a major public health and economic issue. Over the next 20 years, it's projected that the cancer burden is going to increase by 60%. Currently, cancer ranks as the second cause of death in the USA, right under cardiovascular diseases, and there are 18 million cases and 9 million cancer deaths each year worldwide. Over the course of their lifetime, 21% of males and 18% of females are going to develop cancer, and 13% of males and 9% of females are going to die from cancer. So over the past 40 years, there has been a tremendous progress in the early detection methods and improved therapies that has led to the increase of the 5-year survival rate in cancer. As you can see on the top of the plot, for many cancer types such as prostate, thyroid, and skin, the survival rate is well above the 90%. However, on the bottom of the plot, you can see that for many cancer types, such as lung, liver, and pancreatic, they still have a low survival rate, and there hasn't been a lot of progress since 1977. So there is a need for increased cancer research and advanced new therapies and early detection methods. A big issue for this is that developing a new drug is very time-consuming. On average, it takes more than 10 years to go from the bench to the market for a drug. And moreover, most of the drugs fail throughout the different stages of the drug discovery pipeline. As you can see here, the typical example is that from the 10,000 compounds that enter the pipeline, only 5 of them make it to the clinical development, and in the end, only 1 gets approved. The biggest cause of this failure in the drug development is efficacy, with more than 52% of the drugs failing due to low efficacy. Of course, there are other reasons such as safety, strategic, and operational reasons. On the positive side, though, there has been a tremendous progress in new biomedical technologies that has enabled the generation of unprecedented amount of data. As an example, for example, the cost of sequencing has been dropping dramatically over the past years. 20 years ago, it costed more than $100 million to sequence your genome, but now you can sequence it with less than $500. In addition to the lower cost of sequencing, there is a large collection of new high-throughput assays that are constantly being developed and produce different diverse types of data, such as data about the proteome, the genome, and the imaging of the cancer tumor, and thus giving us a more comprehensive information about cancer. All of this big biomedical data can then provide us with the necessary infrastructure to leverage and use AI methods and applications through diverse areas within medicine. For example, we can leverage this big data in the preclinical research and use AI to identify new targets, new compounds. We can use machine and AI methods in the clinical trials to better stratify patients. In the patient care, for patient readmissions and patient intake reasons. And of course, AI plays an important role in the diagnostics, such as in the fields of dermatology, radiology, and pathology. So our center is one of the NCI designated cancer centers. We're here at Florida, and we have 97 cancer research labs that are working to advance cancer research. These labs are part of 3 multidisciplinary research programs, and there are 9 shared resources that are using state-of-the-art facilities and instruments to generate data for all of those labs. So the typical researcher at Sylvester at the University of Miami utilizes these shared resources, uses the state-of-the-art instruments, and produces large-scale datasets. And of course, as we know, there's different levels of processing of the data. The data need to be processed from raw to higher levels of processing. They need to be managed, and this will enable us then to model them. And in the end, the goal of every researcher is to follow this pipeline and reach some actionable insights, such as develop new biomarkers, new compounds, or new precision therapies. So apart from having state-of-the-art facilities and instruments, for us it was essential to also have a robust and scalable data infrastructure that would enable the researchers to not only produce data, but also manage their large-scale and diverse data. For this, we created the Sylvester Data Portal, which is a versatile multi-omics research platform. So the portal is a data repository where researchers store all their data safely and long-term. It's a data management portal where the reagents, the most important reagents, are registered, well annotated, and can be used later on. It's a data analytics portal that gives researchers the ability to not only access their data, but explore them through the UI and also download them for downstream analysis. And it's also a collaboration hub that enables the exchange and the transfer of these big data across the University of Miami ecosystem. One example about the data repository that we tried to solve was the typical use case that when a researcher does a cancer experiment, they produce a few samples, experimental samples, and then they send it to the right appropriate shared resource. And this, of course, generates various diverse data types. So you can have the metadata of your experiment. Of course, the instrument generates the raw data. You have some QC files. And what used to happen was that all of these different files were saved in different formats. So the metadata could be saved in the lab computer, the raw data in the cloud portal of the sequence provider. There were files in the emails. There were files in the postdoc labs. And as you can imagine, when it was time to collect all the data and either publish the results or submit a clinical trial application or a patent application, many of those key and important files were lost or were very hard to find. So that was one of the use cases that Sylvester Portal saves. So it provides the structure to save and securely store all the necessary data from an experiment. The Sylvester Data Portal also is a data management portal, which we ensure that the data sets that are generated through Sylvester are fair, so they're findable, accessible, interoperable, and reusable. The key reagents are registered across Sylvester, so if the same experiment has been used in different shared resources, we're going to be able to track it. We have standardized metadata and data specifications, such as experimental, QC, reagent metadata that helps us standardize processes and the experiments throughout the campus. And also we have publication-ready datasets. That means that from the start of the experiment, we have a checklist and we're collecting all the necessary information that are needed to publish. Also, the Sylvester portal is a data analytics portal. So we really wanted to enable the researchers to have a glimpse and also explore their data. For this, we have a UI-based analytics tools platform that we have widgets that enable researchers to really see their data on the user interface. And for this, we are using the most established bioinformatics tools and the most frequently used tools. We also have AI-ready datasets. Not only did we want to accommodate a UI method of analysis, but for our computational biologists, we're saving all the data in a standardized format. So every assay has its own standardized format, and that can increase the ease that the computational biologists can integrate their data in a downstream analysis. We are also integrating with publicly available data and tools. So researchers not only are going to be able to access their own data that they generate at Sylvester, but we're also importing high impact and valuable data sets from national and international research consortia. Another aspect that we thought it was very important for the portal was to enable researchers to have automated processing pipelines. So for pipelines that are well established and frequently used, researchers would automate the whole process and go from the raw data to the processed data in a matter of a few hours. And of course, throughout our development of the portal, we had emphasized the containerized code base, which will allow for the reusability and reproducibility of the workflows. The last feature of the portal is the collaboration hub. And for this, we had a secure permission and authentication system that would enable researchers not only to share their data, but have different functionalities depending on the roles. So depending on the role of the University of Miami researcher, you have different UI elements and different portal functionalities. You can imagine that, for example, the administrators or the shared resource members would have new functionalities such as to have the overview of all the experiments. All our data have different privacy levels, so the researcher is the owner of their data and they can choose if they want to keep them private, share them with a few collaborators, or also make them public to support their publications or their papers. And of course, naturally, we have different permission levels that, for example, some interns or students, they have only a read-only or analyze permission. So all of these functionalities are included in the Sylvester data portal. It's currently in the public beta version, and it's accessible to all University of Miami researchers. But of course, in the next year, we're planning to make it public and accessible to the general public. And the second now topic that I would like to talk is that once we have these well-organized, curated, and annotated data sets, give you a few examples of how we can now use and leverage machine and deep learning approaches to accelerate cancer research. And the examples I'm going to talk about is through our group, our System Drug Discovery group at the University of Miami, which we're leveraging state-of-the-art approaches to proceed. The first example I'm going to share with you tackles the issue of the large chemical space. So the chemical space is estimated to contain at least 10 to the 60th organic molecules. The bar plot here shows you the number of small molecules that you can order currently from the big providers. So enamine, for example, you could order today more than 3.5 billion molecules. And to put it into perspective, it's more than double than the stars in the Milky Way galaxy. As you can imagine, like testing all those molecules or even screening them, it's not practical. So we need methods to prioritize in silico, computationally prioritize and screen those molecules before you even go into the lab. To this extent, we created our multitask deep learning architecture, KNet. And that came to be based on the aggregation of two large-scale databases, the Campbell and the Kindness knowledge base, which led us to having a robust data sets of 326,000 compounds and their respective annotations and activities for 342 targets. And through our architecture, our multitask deep learning architecture, we then were able to make predictions for all the compounds and extend it to the largest library of enamine and essentially predict based on the structure of the compound if the compound is going to be inhibiting those 342 targets. And this is one example that you can use to prioritize and pre-screen these large compound libraries. A second issue that we tried to utilize and tackle was that currently 75% of research is still focusing on 10% of the genes. And as an example here, the human genome has around 20,000 genes. And an estimate is that about 3,000 of those are druggable. So there are compounds that could inhibit their activities. And the relevant one for our diseases it's another 3,000 genes. And as you can see, what we're interested in cancer research is the intersection between those two sets. So there are about 600 to 1,500 genes that are both disease relevant and druggable. However, there's been this emphasis that the researchers are only focusing on a smaller amount of targets. And as you can see, for example, there are approved drugs for only 600 of those genes. And to this extent, there was created this NIH-funded consortium, the Illuminating the Druggable Genome. And the idea is to promote and identify and extend the research to the rest of the druggable and disease-related genes. Using our tool, we proceeded to do exactly that. So we created this clinical kinase index, which enabled the exploration of these currently understudied druggable proteins. Again, the start was well-curated data that was supported by our data infrastructure. So on the left, you can see here the different databases that we collected data from. In the middle of the graph, you can see different metrics that we passed the data to assess the relevance of the kinases to that particular cancer type. And the end product is a prioritization algorithm that prioritizes the understudied proteins per cancer type. So a researcher can enter their cancer type of choice, and then they're going to see a prioritization list of relevant but understudied proteins that then he can use for downstream analysis. And the last example of our approaches was trying to address the issue of lack of efficacy. As you saw before, most drugs fail the drug discovery pipeline due to lack of efficacy. A solution to this problem is to develop combination therapies that can then advance and progress the efficacy of the drugs. In our links consortium that we're part in, we're having this connectivity map approach that says briefly that when you have a disease signature, that you know that these genes, for example, are upregulated or downregulated in the disease, the optimal drug would reverse part of the signature. And in our research, we wanted to go into a step further, so we wanted to identify two compounds that they are orthogonal and they're reversing different parts of the disease. And to this, we created a synergy-seek algorithm that again started with the aggregation of more than 1.2 million data points that included more than 1,700 compounds in 73 cell lines. And also we aggregated RNA sequencing data from 156 tumor samples that were publicly available. And then the next step was to correlate both the disease signatures and the drug signatures using our algorithm. And the end product was to create a prediction algorithm that would predict the compound combinations that would be efficacious for that cancer type, and therefore enable increase the efficacy of drugs. So as a summary, we saw today that we've had progress in both the data infrastructure and then analysis of cancer research. So through our portal, we're enabled researchers to analyze their data, access them, share them, and manage them. And these well-curated now and well-annotated data sets can be used in downstream analysis, leveraging established or new machine learning and deep learning algorithms, such as the one I showed you, that the goal is to prioritize compounds, prioritize drug targets, and prioritize therapies. And for this, of course, I would like to thank our Sylvester data portal team, which in a very short period has developed the portal, our oncogenomics resource, our system drug discovery group, the whole department of pharmacology at the University of Miami, and our link center, which we are part of as the data integration and coordination center, and of course Microsoft for giving us support for the portal. And I will be happy to take any questions you have at this point.
That was presented by Vasileios, Stathias lead data scientist at Sylvester Comprehensive Cancer Center, who provided insights on data science strategies applied to management and analysis of large-scale cancer data sets. If you enjoyed this video, make sure to subscribe to our channel and find more presentations like this just below.